A monohybrid cross is a cross that involves one gene, or in other words, we're just looking at one gene at a time. So here in this example, we are looking at the B gene, and we're going to start with two true breeding parents. And so by calling them true breeding parents, we are saying really that both of them are homozygous. So this one over here, we would refer to that parent as the homozygous dominant. And this parent over here, we refer to it as the homozygous recessive. So when we cross these two with each other, the first thing that has to happen really is we have to get meiosis in both of them. And remember that meiosis is really producing the gametes. So we're going to need an egg from one parent and we need a sperm from the other. So if this first one over here, if I make this mom and I make this dad over here on this side, mom is going to undergo meiosis so that we can get an egg. So here's going to be my egg from mom. And in the egg, we are going to have a capital B. And that's because mom is homozygous, which means she only has capital B's there. So no matter what, that is what we're getting in that particular egg. From dad, we're going to get the sperm produced. And dad is also homozygous. So we know that that sperm is going to get a lowercase b because that's all that he has. So it's really easy to come up with the gametes when we have homozygous parents. Okay, so those are our two combinations. Now the next thing is going to be really fertilization. So we're going to get both of these combining together through the process of fertilization. And that will result in really fusing those two together or combining them so that we get a single cell that contains whatever was in the egg and whatever was in the sperm. And that is now going to be what we call the zygote. The zygote itself is, notice that it's going to be diploid, so it's got two alleles in it, and in this example, it is going to be heterozygous. Now we haven't talked about phenotypes yet, we've only talked about genotypes. So if we wanted to go back and let's actually assign some phenotypes to this. So I'm just going to make this a generic color gene. So we have two forms, we have capital B and I'm going to call that black. And we've got lowercase b, and I'm going to call that brown. Okay, so mom over here is going to be black or have black hair or black something, and dad over here is going to be brown. Okay, and that would be the case because mom has two copies of the capital B, which is black, and dad has two copies of the lowercase b, which is brown. Now, when we look at the zygote, we can assign a phenotype to the zygote as well. But since we have at least one capital B here, we know that the zygote is going to also be black. Okay, so that one is going to be just as black as the mother was. We wouldn't even be able to visually tell that there is information there in that individual for making brown in the presence of the lowercase b. Another thing that we can do is we can name the different generations that we have. So if we wanted to go through and name them, the parents up here at the top, they form what we call the parental generation. And the parental generation, we just designate with the letter P. Okay, so that's the P generation. The next generation is what we refer to as the F1. And F1 stands for the first generation. Now, if we wanted to go on and we wanted to produce the next generation is going to be the F2. And then following that would be the F3 and then the F4 and so on. But we always use the previous generation as parents for the next. So what we mean by that, if we want to produce the F2 generation, we need to use two F1 individuals to serve as the parents for that. So let's go through and let's look at if we take two individuals that have this particular genotype to them. So we're talking about this heterozygous big B little b. We're going to take two of them and we're going to cross them with each other so that we could get that F2 offspring. Okay, so up here at the top, I am writing my two F1 individuals that I'm going to be crossing with each other. And I'm using, notice, the F1 as parents to produce the F2. Okay, so we're really trying to produce the F2 generation. Now, just as we saw with the previous parents, that P generation, when we crossed them with each other, the first thing that had to happen was meiosis. And that's the case here as well. So when we take one of these parents, when they go through meiosis, if we just look at this one over here, we would expect that half of the time the eggs are going to get a capital B in them, and half of the time they're going to get a lowercase b in them. And that's because we have two different alleles present in that heterozygous parent. And so we really don't know which one of those two is going to be the randomly chosen egg for fertilization purposes. So let me just label that these are eggs that we're looking at here. Now from this other parent that's producing sperm, we have a similar situation because it is also heterozygous. So we expect that half of the sperm will have the big B and half of them will have the lowercase b. And so this becomes a little more complex because when we get fertilization, we don't know which one is going to get chosen from either side. So we could get the capital B over here chosen, and maybe we get big B over on the other side as well, or maybe we get this combination, or perhaps this combination or this combination. So now we have a number of possibilities that could occur. And this is where we use a tool that's called the Punnett Square. So the Punnett Square is named after the individual that first described it. And this is going to be a tool to help predict the possible offspring. Okay, so when we draw the Punnett Square, what we're going to do is we'll just start with two sides. That's the way that I do it. And we're going to put one parent's possibilities down one side. So I'm just going to put mom on this side. And then we put the other parent across the top. And so I'm going to put dad up there. But what we're going to put on the side is going to be gametes. And so since it's gametes, this is really, instead of putting mom, you could say this is where I'm going to put my eggs. And over here, instead of writing dad, we could say this is where I'm going to put my sperm. Okay, so that's what we're going to put on here. So half of our eggs are going to have a capital B, half of them will have a lowercase b. And half of the sperm will have the capital, half of them will have the lowercase. And then what we do is we just divide this into a number of boxes. Okay, so hence my square that I end up with. And then all that I do to fill it out is I look directly across and directly above, and I write those two letters into the square. So the first box is going to get two capital B's. This one here is going to get a capital B and a lowercase b. This one is going to get a capital B and a lowercase b. And this one's going to get lowercase, lowercase. Now, if we talk about what we have inside of these boxes, inside the boxes represents the possible offspring. Okay, so all of this is possibilities. So my offspring could get a number of different genotypes depending on which egg and which sperm was randomly selected. So let's talk about these possibilities that we see inside the box a little bit more. So every one of the squares, and we've got 4 squares, represents a genotype. So if we go through and we list the different genotypes, we can have capital B, we have a possibility of heterozygous, and then we also have the homozygous recessive, which is the 2 little b's. If we went through and we counted up how many we have of each one, and that's all I'm doing right here, what we get is what we call the genotypic ratio. So this is a ratio of the genotypes, what we expect to be possible. If we wanted to ask, taking this cross, what is the probability of getting an offspring or getting a child that is little b, then we would simply count up how many boxes we have that represent that genotype, and there's just one, and then we would divide it by the total number of boxes that we have. So the probability of a child with little b, the answer to that would be 1/4, because we just look here and we count up how many boxes match that particular genotype, and again divide by the total. So that's the genotypic ratio. We could also talk about the phenotypic ratio. And remember that the phenotype is the visible appearance or the looks. To do that, if we go through here and we talk about what each one of these offspring looks like, as long as they have at least one capital B, they're going to be black according to the colors that we assigned earlier. So we actually have 3 boxes that are going to show black, and then we have just 1 box down here that's going to show brown. So we can come up with a phenotypic ratio as well. And to do that, we would write down the different phenotypes that we have. We have black and we have brown. And then we're just going to add up how many we have. And we have 3 black and we have 1 brown. So that right there would be our phenotypic ratio. If we wanted to ask a question and say, what is the probability of a black offspring? All we have to do is go through and add up how many boxes are black, which in this case is 3, and we divide it by the total. So that would give us 3/4, or you could say 75%. So when we're asking for the probability, we're looking for either a fraction or a percentage as our answer choice. And that would be a look at a very simple monohybrid cross. Now another thing, all of these offspring that we're seeing inside of this box, that would be the F2 generation. Okay, so don't forget that the F2 generation is what we produced here, since we were using F1 individuals as parents.